So as we continue our look at the pathway of digestion, we'll entitle the next flowchart, Digestive Pathway 3. And what we stopped before in the previous flowchart on was the stomach. We completed our look at the stomach, and now let's reiterate and sort of summarize what happens after the stomach. After the stomach, you are going to have a lot of stuff being digested, right? We have these enzymes within the stomach, we have that pepsin, we have the gastric juice. All of that is going to affect the bolus that entered the stomach. So there are going to be several boluses, or bolusi, whatever you want to call them, that enter the stomach and basically get churned and digested. That's going to result in food as a whole turning into a new substance that we're going to call chyme. Okay, so chyme is the result of the stomach doing its digestive job. And so here, food is now referred to as chyme, and chyme, because it's coming from the stomach, has a pH of 2. So it's a very acidic component that's the result of stomach secretions and digestion. Now, we can state that chyme is basically going to be a very, very soupy mixture. That's the sort of consistency that it has. It's a soupy mixture of a couple of different things, namely partially digested carbs, okay, partially digested carbs, and that's because at the mouth, if we remember, we had some sort of carb digestion via salivary amylase. Also, it will be a soupy mixture that contains smaller polypeptides, not amino acids, just smaller polypeptides, because the stomach itself was able to activate pepsin in the presence of HCl to give you those smaller polypeptides. And also, there will still be some undigested material here. So there will still be some undigested material. So what you can really summarize and state about the stomach is that though it involves itself in a decent amount of digestion, not all digestion occurs in the stomach. The majority of digestion is actually going to occur in the next part of this pathway. So what we're going to do is first label how we get there. It's through a sphincter, from one compartment to another. So the chyme, therefore, moves through a sphincter, and that sphincter is known as the pyloric sphincter. And you have to ask yourself, is this movement voluntary or involuntary? It's of course involuntary because the pyloric sphincter is the muscular valve that is composed of smooth muscle. So it moves through pyloric sphincter to the next structure, the next compartment, known as the small intestine. And this is where a lot of digestive action is going to occur. Let's take a look and begin by now labeling the fifth component of the digestive pathway as the small intestine. So I think what's first important to recognize is why it's called the small intestine. Well, first of all, the small intestine is very much not small, depending on which way you look at it, because it's actually 6 meters long. So why is it called the small intestine? Well, this is because it's small, let's say in quotes, depends on the context. It's because it has a smaller diameter than its counterpart, its large intestine counterpart. So versus the LI, which is the large intestine. That's the only reason it's small. It has a very long structure, 6 meters long, tons and tons of folds. That tells you something about its structure, that it has a very high surface area to volume ratio with lots and lots of folds. So it's a very, very elongated structure, just a small diameter. In addition, the small intestine contains 3 major divisions. And these are 3 divisions you should remember. The first division is the one that's, I think, most important because this is where we have the most digestion. Duodenum is our first division. This is a major, absolutely major, I would say majority site of chemical digestion. Lots of chemical digestion will occur in the duodenum because we still have a lot of stuff to break down here, as you saw, because chyme is going to enter the small intestine through the pyloric sphincter. So we got to break this chyme down even more. The duodenum is going to be a major site of that breakdown. The second division, in order of where the small intestine is going to be, the second division is the jejunum. Very fun to say, jejunum. And then the third division is going to be the ileum. And the last division is going to, of course, lead into the next compartment via a sphincter, which we'll see. So those are our three divisions of the small intestine. The small intestine is very much going to be characterized by a large surface area. It has lots and lots of surface area, and this is because of its structure. I would say its cellular structure specifically, because it has lots of what are known as tiny villi, tiny fingers, finger-like projections. Many of these are going to be spread throughout the 6 meters of the small intestine. And so what we see with this microvilli structure is that they present themselves as membrane folds, and we know that anytime you see this word fold, you're going to be talking or increasing surface area. Membrane folds on what is known as the open small intestinal epithelial cell surface. So this just means that on the outside portions of the small intestine, what you're going to have are these finger-like projections that are going to have very direct access to whatever's being put into the small intestine. They're going to interact with this chyme in a great deal. And the reason why we have this very, very intimate interaction and direct interaction is because this is overall, if you increase surface area, remember fungi, and we always said that they have a high surface area to volume ratio because of their mycelium and hyphae that extend out. It was all to do what? What's fungi's number one job? They're very good at absorbing. The small intestine, therefore, is also very good at absorbing because of its large surface area. This all is going to allow for lots of absorption. This is the major site of absorption after we have enough of the chemical digestion that occurs. Okay? So after we have this major chemical digestion, we need to absorb it into the blood and then therefore into the cells. Remember, food processing, ingestion, digestion, absorption. Absorption will happen primarily within the small intestine for this reason. Now, how can we get this major chemical digestion to occur? Because we need to do that before we get to the absorption. This is done via something known as pancreatic juice. So pancreatic juice is a material that is created and produced at the pancreas, but then sent to the small intestine. Pancreatic juice arrives. Okay? It specifically arrives at the duodenum primarily. So, when pancreatic juice arrives and comes from the pancreas, it contains within it a lot of stuff. Things to highlight are that it has bicarbonate ions all throughout it, and this is just known as bicarbonate. If you want to talk about the actual term, HCO3- is bicarbonate. This is a very, very basic compound, meaning that it has a very high pH. The structure that came into the small intestine, the stuff, I should say, that came into the small intestine had a very low pH. It was very acidic. If you combine something that's very basic with something that's very acidic, you get a neutral compound, and that's exactly what we're trying to do. Because remember, the stomach is the only thing really in the body that's capable of handling pH 2. Now we have to put it back to the normal neutral sort of pH, and that's what we'll do. We'll just flood it with some bicarbonate ions. This is going to overall give you a high pH plus a low pH, which means a medium pH, which is a neutral pH. This neutralizes the acidity of the entering chyme. So chyme is now neutralized. We can now work with it. We can use some other enzymes that are going to be a part of the pancreatic juice, because the pancreatic juice also contains many enzymes. We'll talk about which enzymes specifically when we actually talk about the digestion of specific molecules in a later flowchart. But just know for right now the pancreatic juice contains many hydrolytic enzymes that will do tons of digestion. And of course, when we need to get the pancreatic juice to the small intestine, this is going to be through a duct. A duct is going to directly carry the PJ, pancreatic juice, to what part of the small intestine? The part that is very good at chemical digestion, the duodenum. This would, of course, mean that this is an exocrine secretion coming from the pancreas. So, overall, this gives us the theme that there's lots of digestion here, lots of absorption here, all at the small intestine. Final thing about the small intestine that we'll do up here is the fact that it also will involve itself in this structure called bile. Bile is a structure, is a substance that is produced in the liver, so it's not produced in the small intestine, it's produced in the liver, and it's actually stored in the gallbladder. Okay, stored in gallbladder. So those are both structures that are not the small intestine. Why are we mentioning it then as a part of the small intestine? This is because bile will get to the small intestine, it will get there and arrive there via this process. It passes through the bile duct. Again, another duct. Lots of ducts in digestion. Passes through the bile duct to the major part of the small intestine, and that is the duodenum. Okay, so that's another very important part. It's always going to be lots of chemical digestion and absorption here. Passes through the bile duct to the duodenum. Now, what's the big deal? What is bile? Bile contains structures, substances called bile salts. So what are bile salts useful for? Bile salts are good because they break down, physically break down, they mechanically digest fats. Mechanically digest fats. This is the first time we're mentioning fats, so it's important to recognize that fats are really not digested at a great deal only until they reach the fifth component, the fifth compartment. So they take a long time to get digested. So this happens at the duodenum of the small intestine because of the bile salts. Bile salts basically break large fat globules into smaller fat globules. That's mechanical digestion. Please note that there is no digestive enzymes involved in this process. Bile salts do not use them, therefore there is no chemical digestion of fats as of yet. Only mechanical digestion of fats. That covers our look at the small intestine. We now have gone from the oral cavity, then we went to the pharynx, and then we went to the esophagus, then we went through the stomach, now we're at the small intestine. The next step is, of course, now to go through the ileum into the large intestine.